A very good day to everyone. My name is Ashreen. My supervisor is Associate Prof. J. Hazrul Hanim and also Dr. Ahmad Kamil as my co-supervisor from Lembaga Cocoa Malaysia. I'll be presenting a little bit about luminescent nanoparticles and how I produce it from cocoa pod husks and how its potential to be used as bioimaging tools. The luminescent nanoparticle in question is carbon dots. So what are they? They are actually miniature spherical balls and they are only about the size of 6 to 8 nanometers. They are very small. They can be known by many names actually. Carbon nanodots, quantum dots of graphene, or quantum dots of carbon. It actually has been made from various natural precursors such as Manilcora zapota, guava leaves, and Syringa oblata. So there's many more that have been used actually in literature. So why I did this is because since they are using natural precursors and Malaysia is abundant of this, I think that Malaysia has a very good potential for nanomaterials. So therefore, Malaysia is blessed with abundance of agricultural products. Ergo, we have plentiful of agro waste. So thus, we have a huge potential of sustainable nanomaterials production. So the many applications of carbon dots so far in literature that I found will be radiotherapy, phototherapy, drug and gene delivery, bioimaging that I'm going to show you, antiviral, antimicrobial, biosensing, and antifungi. So the production of carbon dots, there are actually like two ways. One is the top-down methods, and the other way is the bottom-up approaches. So first we will see the top-down methods. The three main top-down methods that I'll be discussing would be laser ablation, arc discharge, and electrochemical. So laser ablation, it aims macrocarbon such as solid carbon or graphite with laser energy, and it severs the bonds and removes the carbon dots present on the surface. And also arc discharge, it applies electrical charges between two graphical electrodes and then it gives off pieces of small carbon known as the carbon dots. And then we have electrochemical, utilizes the electrolytic processes. Electrodes are usually larger organic molecules such as the graphite rods. It's a one-step process. In comparison to bottom-up approaches, top-down method requires a lot of energy and sometimes chemicals. So therefore, bottom-up approaches seem to be more preferable by most researchers. So now we'll go to bottom-up approaches that have been used so far. So the bottom-up approaches would be microwave, hydrothermal, and pyrolysis. So microwave energy is used to disintegrate the chemical bonds, cost-effective and low energy utilization. Hydrothermal precursors are grinded into smaller pieces. Water can be used as solvent, and the organic compounds are transformed into carbon dots. High temperature, high pressure, cost effective, also one step. Pyrolysis, in the presence of acid, alkali, organic precursors are subjected to high temperatures. It depends on the researcher whether they want to eat what type of acid, what type of alkali, and how high the temperature is. It's also simple and it's cost effective. Since bottom-up approaches are much more simpler, it's only a one-step synthesis. Therefore, I use the microwave method to produce these luminescent nanoparticles. The material that I'll be using in my research would be the cocoa pod husks, which comes from Lembaga Cocoa Malaysia. Thank you very much. So, cocoa pod husk is known as Therobroma cocoa, and so it is commonly planted in the Americas, Asia, and Africa. So the cocoa industry generates a lot of waste and mostly constitutes of this cocoa pod husk, because cocoa pod husk is like 75% of the fruit, and annually Malaysia actually disposes about 923,000 kg of CPH. So that is actually a lot. And CPH has various chemical components. For example, they have crude fiber, crude fat, cellulose, hemicellulose, lignin, pectin. They even have enzymes such as lipase. So moreover, in the phytochemical screening, polyphenols like tannin, alkaloids, and saponins have been found to be contained in the cocoa pod husk as well. So these polyphenols are important in the protection against degenerative diseases like coronary heart, cancer, and neurodegenerative disorders. It also has a protection mechanisms. It has microbial activities. So so according to Adidaco, they actually did an experiment, and it showed that cocoa pod husk actually showed antimicrobial activities due to the polyphenols. These polyphenols actually penetrate the bacterial cell wall, and this is why I use cocoa pod husk as a natural precursor to develop these luminescent nanoparticles, in hopes that whatever attributes that this cocoa pod has, it will be translated into environmentally friendly and advantageous luminescent nanoparticles. Okay, so moving on to materials and method. So I only use a one-step microwave synthesis. So I'll take the cocoa pod, I'll dry it, I'll grind it, and then I'll mix it with just water. No chemicals are involved in my method. So, and then I'll put it in the microwave irradiation, and then after that I'll let it cool, and then I'll centrifuge, and then I'll filter it with Whatman filter paper, and therefore I get the luminescent nanoparticles. As you can see, it is green in color. So I did characterization by means of Fourier transform infrared spectrum, FTIR, UV's absorption, VV's photoluminescence spectroscopy, PL. To test the cytotoxicity of my luminescent nanoparticle, I did brine toxicity assay. So I want to make sure that if it's going to be used as a bioimaging tool, it does not inflict any danger onto the cells. So under how it was is that brine toxicity assay is practically like I want to evaluate the cytotoxicity of the CPH based CDs. Our model organism is Artemie nauplii, small little shrimps, they move very fast. Each well, I put at least 10 to 15 eggs. So they were incubated with the carbon dots for 24 to 48 hours, and then I count its hatching percentages, like a number of hatch over the total, then times 100%. Lethality is like number of death over total shrimps times 100%, mostly after 24 hours, and LC50 would be the median lethal concentration. Okay, so these are the results that I'll be going to be talk about. So after I successfully synthesized the nanoparticles, I ran it through FTIR first, and I discovered that under FTIR there was a peak observed at around 3200 to 3300, okay, which is indicative of the vibration stretching of the OH-NH bond. So you can see that this actually stipulates that carbon dots are hydrophilic, and also there's peaks around 1725 and 1040, which corresponds to C double bond O and C single bond O stretching vibrations. So also the functional code like C double bond O shows the stretching results and the presence of other carboxylic group. So when the other carboxylic group, it means that there is a presence of oxygen on the surface. So when there's an increase in oxygen, it leads to an increase in surface defects. When there are defects, these excitons will be trapped, giving a rate-shifted emission. So the peak at 2920 would be related to vibrations of CH stretching. So like again, I reiterate, presence of these functional oxygenated groups, as you can see. All the oxygenated groups will give rise to soluble properties of the carbon dots. So all FTIR features contribute to the outstanding solubility of CPHCDs. So it'll be very useful for further modifications and biological applications. Next, to UV's absorption. UV's absorption was to show that the synthesized nanoparticle is really what it is. It's like it is a indication that I have synthesized the nanoparticle successfully. So we had like the sharp peak here at 235 nm due to the C double bond O due to the n-pi electronic transitions, and also a broad peak at 293 nm, pi electronic transitions, and CC double bond. So the presence of this electronic transition proved there is an occurrence of functional groups on the surface of the CPH CDs, and also that the findings showed that the specific peaks indicate the successful formation of CDs by just using a very simple microwave method. So now I move on to photoluminescence. So when I illuminated it by UV light, the echo solution of the CPH based CDs, as you can see, was green in the picture previously. So the PL spectrum showed that PL emission wavelength of CPH CDs. So at 380 nm and above, something very weird happens. So I start at 300, so it's excited for every 20 nm, and above about around 380, they start having twin peaks. I think this is a very maybe it's because from cocoa pot Husks, and it has this very unique signature of photoluminescence. And these peaks, actually the maximum emission peak is at 490 mm when excited at 400 nm. So you can see here, right? So these twin peaks actually overlap each other until 500 nm. Okay, and so overall the trend shows that by increasing the excitant wavelength, the peak turns to redshift. So here, so it goes to redshift due to the photon reabsorption. So the twin peaks actually reappear, and it overlaps at 400 nm and so on with excitement. So this shows that the nanoparticle is excitation dependent, and also that by referring back to the FTIR results, when you have the presence of the C double bond O and other carboxylate groups, it increases the presence of oxygen on the surface. So the increase in oxygen group tend to lead to a surface defect. So therefore, that's why we have the rate-shifted emission. So therefore, then we go to brine stream lethality assay. As you can see, after 24 hours, the lethality percentage will increase. Okay, brine stream had a better survival rate compared to the standard solution, which is this. Okay, the standard solution is just salt water. So at 0.001 milligram, it shows lower lethality lah, at 57.93%. The lethality percentage was 61.46% in comparison to the standard solution. It was observed the brine streams that hatched were very active. So after 24 hours, they weren't they didn't survive so well because there was no oxygen or nutrient. So this is LC50. So it shows that from this linear regression, I got 1.32 milligram per ml, meaning that it is, according to Mayer's toxicity index, the LC50 is if it's less than 1, it's considered toxic. So in my case, it's 1.32, meaning it is not toxic. And the hatching percentage would be the highest hatching percentage, as you can see, is in 0.01 compared to the standard solution. So it is here 0.01 followed by 0.01 and 0.1. So at 1 milligram per ml after 24 hours, it has the lowest hatching percentage. So at 1 milligram ml, it doesn't hatch so well. It hatches, but it hatches a lot, but it gets stuck. So I'll show you a picture of it. See. So from figure left to right shows brine streams in different concentrations, 0.01, 0.01 and 0.1. So this is 1 milligram. So the streams were observed to have slightly disintegrated by high concentrations of CPHCDs. Here, this is a high concentration, and this could be due to the persistent presence of Rhizopus species fungi isolated from the cocoa pod in a different study by Jimet et al. So as you can see, until 0.1, it has very good survival after 24 hours. So meaning that that means that the cocoa pod Husks actually include nutrients that induce the seeds to hatch. So however, once hatched, the brine streams either live healthily or are suspended in their umbrella phase. This is the umbrella phase, depending on the dosage of the cocoa pod Husks carbon dots. So if you can see here, So in addition to that, so a small dosage is sufficient for biomedical applications to be used. So it'll be very convenient for the body to effectively excrete nanoparticles, because we're only using nanomaterial 0.01 milligram per ml, and until 0.1 milligram should be enough to do for bioimaging tools. So in conclusion, it is summarized that CPH, a biomass waste from the cocoa industry, can be most economical and readily available precursor for carbon dioxide synthesis by means of an easy microwave irradiation method. So it was also discovered that the as synthesized green carbon dots from cocoa pods do not cause toxicity and are safe to be used for bioimaging and drug delivery in the medicine sector. So thank you. That's all my presentation. These are my references. This is my references.